So now that we've covered basic skin anatomy, let's talk about how skin develops during embryonic life. And of course, the big question is how do we get from something like this, around 4 weeks embryonic, 4 embryonic weeks, to a fully developed organ, the skin? All right, we'll start by talking about the epidermis. So when you're forming the epidermis, the surface ectoderm is going to differentiate. So the epidermis is derived from the ectoderm. It divides into two layers, the periderm and the basal layer. Now the periderm is going to be your outer or more surface layer, and the basal layer will be the inner layer. Now the periderm is not long lasting. It's gone by the end of roughly month 5 of fetal life. But the periderm is shed into the amniotic fluid, which makes it part of the sample that you collect when you do a procedure like an amniocentesis. And so for some babies, those cells are actually of critical importance because they're part of a testing process. But ultimately, they don't make it into the final skin structure. Now after that, around embryonic week 11, the basal layer will further differentiate into an intermediate layer, which will be composed of keratinocytes, and then what the book is calling stratum germinavatum, which is really the stratum basale that we talked about in our last lecture. From there, around week 20 of embryonic life, the intermediate layer will further differentiate into the layers that we discussed in the skin anatomy, the stratum spinosum, stratum granulosum, and stratum corneum. Now this process of differentiation starts cranially, it starts at the head, and then it sort of moves caudally. Okay, so it starts at the top and moves toward the bottom. And that becomes relevant when we think about the paths that future skin cells take on their way to their final locations. And in fact, those pathways are well defined and they follow the patterns of neural crest cell migration, which makes sense. Those lines, those paths that the cells follow are called the lines of Blaschko. I believe Blaschko was the physician who discovered them. But what we see is that if there are anomalies of the skin, sometimes we see those anomalies following those lines, particularly if the skin defect arises during neural crest cell migration. So we can get something that looks a little bit like this. So where you see, in this case, it's a pigmentation issue, particularly sometimes sex-linked pigmentation issues, because then you get dual expression in females but not in males. And they follow X activation patterns, which is something that will be covered in more detail in another class. But this is the idea. You can see how the darker patches on this person's trunk match the lines, the patterns of neural crest cell migration. This would be somebody who has a sort of a segmental expression of this particular phenotype. You can also have it affect the entire body, and in rare cases, you may actually be able to see all the lines. All right, epidermal cells, where do those come from? So the cells of the epidermis, and we had talked about the multiple types. In this case, we're going to look at the melanocytes, the Langerhans cells, and the Merkel cells. Now remember, the melanocytes are your pigmentation cells. So those are the ones that help control the color of the skin and the hair. They are derived from neural crest, which is part of why you see that patterning with the lines of Blaschko, because if you're expressing differential amounts of melanin in your melanocytes, then you may see different populations of cells migrating with that sort of different expression, and then you get that sort of striping effect of lighter and darker skin tones. Regardless, the melanocytes are present in an embryo by about embryonic week 6. Some of them will associate with hair follicles to produce pigmentation for the hair follicles. They do both skin and hair. And in fact, they make up to 10% of the epidermal cells in an adult. Now, the Langerhans cells are more about immune response. These cells arise in the bone marrow, and they continue to arise over the course of your lifetime, so it's not just during embryonic life. And they're present by embryonic week 7. Finally, we have the Merkel cells. Remember, those were associated with neurons. They're involved in touch, pressure, pain, sensation. They're derived from the epidermis itself, and they arise between months 4 and 6. And they are actually connected to the neighboring keratinocytes by desmosomes, by cell junctions. Okay? All right, so the dermis. how do we get the dermis? So it brings us back to embryonic week 3 and our lateral plate mesoderm. So the trunk, the dermis of the trunk is actually derived from the lateral plate mesoderm and some of the somites, whereas the dermis of the face and the neck come from the neural crest cells. So this lateral plate mesoderm we saw back in week 1 during embryonic week 3. So if we look back at our final skin structure, so during embryonic week 4, during development of the dermis, you get blood vessels that are forming in the dermis, which will then associate with developing hair follicles. And that'll be important. That'll help provide nutrients as the hair follicles form. Additionally, in embryonic month 3, we get formation of these dermal papillae, which we talked a little bit about before, but they're actually projections of the dermis that sort of extend into the epidermis and then creating corresponding projections of epidermis into the dermis. And that layer of cells there is called the papillary layer. And the ridges are referred to as epidermal ridges or dermal papillae, depending on whether you're looking at the dermis or the epidermis. Those ridges will form visible ridges on the surface of the skin. So we talked about fingerprints as an example, but if you look at the palms of your hands, there are actually what looks like lines or fingerprints across the entirety of your palm, and those come from the formation of these dermal papillae and the epidermal ridges. Now, the thickening of the dermis continues into childhood. It's not nearly as thick when you're born as it will be several years later. All right, so skin derivatives. So these are cells that are derived from the skin cells. Hair, we've talked a little bit about this already. The hair follicles of the face, but not the scalp, form during embryonic month 2, and then all the rest of them form during embryonic month 4. So they're forming over a period of several months during the formation of the baby. However, you do not acquire new hair follicles after birth. So at puberty, when you see an expression of an increase in body hair, those follicles were there. They were just producing very fine hairs and not as obvious. Now, when we form hair follicles, we start with surface ectoderm in the basal layer, and it induces what's called a hair germ, which is sort of a bundle or a group of cells that then project downward to form the hair peg. So they extend into the dermis and they expand. And once they grow to a certain level, the hair shaft then starts to grow outward back up toward the epidermis. Melanocytes, as we talked about, will come and associate with the hair follicle to provide pigment to the growing hair. And in a lot of locations on your body, you'll actually get small muscles called erector pili muscles that become associated with hair follicles, and they're responsible for raising the hairs when you're frightened or cold. Now, hair at birth varies quite widely among infants, and it changes in color, texture, density over the lifetime of a person. But in particular for infants, the hair that they have at birth is not always the hair they have 6 or 9 or 10 or a year later. And anyone who has had a baby will discover that their hair changes a great deal, especially in those early months and years. Now, the hair follicles do cycle, so a new hair shaft grows in, it starts growing in once the old one has been shed, and so you get sort of a cycle of growth for hair follicles. All right, so this is just sort of outlining the basic anatomy of the hair follicle, of a mature hair follicle. And what's sort of interesting, and I wanted to point out here, is this bulge that's slightly separate from the base of the follicle contains stem cells that will actually help to rejuvenate the follicle over the course of its lifetime. Additionally, there are several glands that can be associated with hair follicles, sebaceous glands and sweat glands, and we're going to talk about their development next. Okay, so sebaceous glands, sweat glands, both of them are derived from the epidermis and they grow down into the dermis. In the case of sebaceous glands, they're often associated with hair follicles. In the case of sweat glands, they may or may not be associated with hair follicles, but when they grow down into the dermis, they form this very sort of spiral curved structure. The sweat glands are also usually innervated with muscles, that sort of trigger them to release sweat. So the contraction of the muscles around the sweat glands then leads them to release sweat to the exterior. Okay. Sebaceous glands are responsible for producing oil and often again associated with the hair, and clearly, of course, with the skin. So often the bud from the hair root, often they bud from the hair root, but they can arise separately. So you can have oil-producing glands associated with hairs and those that are just on the skin. However, they're present usually by about month 6 during embryonic or fetal life. They are also responsible for producing vernix. Now, vernix is this very waxy coating that you see on babies, particularly premature babies, that in theory, the theory goes that it may protect the baby's skin in utero, but it's usually gone in full-term babies and certainly in post-term babies. Now, those glands are there in babies, and what's kind of interesting is that babies can in fact get acne, and often do, frequently in response to maternal hormones in breast milk. So in this little baby's nose you can see those small, essentially baby acne, but they're harmless, they don't scar, and they go away on their own. Now, oil production from sebaceous glands is linked to sex steroids, and it's produced through a holocrine mechanism, which means that the secretory cells, so the cells that are creating the oil, actually burst, and that releases the oil to the surface. Now, sweat glands work a little bit differently. There are 2 different types, apocrine and eccrine sweat glands. The apocrine sweat glands, in this case, the secretory vesicles sort of pinch off of the secretory cells, and that releases the contents. So the cells themselves sort of form these little bubbles on the surface that contain the sweat, and then they burst and release without destroying the original cell. That kind of sweat gland is generally associated with hair follicles. In the baby or in the fetus, it covers most of the body, but for adults, we really only have sweat glands in the armpits and genital regions. And they also produce odors, which are of various uses, and certainly there's been a great deal of interest in the idea that humans can be attracted to one another by their odors. The eccrine sweat glands do cover the entire surface of the body. They may or may not be associated with hair follicles. They release sweat in response to temperature changes or emotional changes, and they are critical for homeostasis, so they're critical for cooling, to help the body cool in hot weather. In this case, the fluid, the sweat, is secreted across the cell's plasma membrane. That's the eccrine sweat gland mechanism. So in this case, nothing bursts, nothing ruptures. The fluid is just secreted across the cell's plasma membrane. All right, mammary glands. So in this case, these are the forming of the glands that will ultimately be involved in producing milk for future babies in women, but men have them too. Men have mammary tissue. It's just, there's just not as much, but it's there. So for formation of mammary glands, the epidermis thickens to form these mammary ridges, and then the primary bud forms at the site of each future breast and grows into the layer of fat, and that's what they're showing in the picture here. So we form this ridge, and then it grows inward into the fat, and then we start to get this sort of branching effect, secondary and tertiary budding, which creates this sort of tree-like shape. So it branches into many lactiferous ducts. Those are the ducts that are ultimately going to produce milk after a baby has been born. But they all open into this sort of mammary pit, which then develops into the nipple. Now, usually in newborns, the nipple is inverted but becomes everted at some point after birth, in most but not all cases. Now, in humans, you really only have one paired set of nipples and breasts, but you can get accessory nipples, which are sort of an evolutionary throwback, and they tend to follow along the line, along the abdomen, where you might expect to see extra nipples in a different mammal. Finally, we'll talk about nails. Nails are derived from the same tissues as skin and hair, and so a lot of times when we're thinking about genetic disorders that affect development of skin and hair, they tend to affect nails too. Now, in this case, of course, this presumes that you have fingers developing, which we haven't talked about yet, but we will get to in other chapters. But basically, you get a thickening of the epidermis on the distal end of the digits, dorsal side, so where you would expect nails to be, and it tends to migrate proximally, so towards the fingertips. We get the development of these nail folds, which are derived from the ectoderm, and they're on the sides and sort of proximal on the nail. And they're showing them in the picture here. Now, from the proximal nail fold, the stratum germinavatum becomes, or the stratum basale, depending on which book you're looking at, but at any rate, it becomes the nail matrix, which is composed of these compressed keratinocytes, and they're compressed down to the point where they become very hard, because of course, your nails are much harder than the rest of your skin. And they grow, of course, toward the fingertips, and usually by birth they've reached both the ends of the fingers and the ends of the toes. And newborn babies often need their fingernails clipped fairly promptly because sometimes they've extended past the tip of the fingers.